Sees Topic 3.3 Nutrition and Energy Systems. Annotated diagram of the ultrastructure of a generalized animal cell. Annotated diagram of the ultrastructure of mitochondrion. Define the term cell respiration. The controlled release of energy in the form of ATP from organic compounds in cells. Explain how adenosine diphosphate can gain and lose a phosphate molecule. The ATP molecule acts as a chemical battery, storing energy when it is not needed, but able to release it instantly when the body requires it. ATP works by losing the endmost phosphate group when instructed to do so by an enzyme. Conted. This reaction releases a lot of energy, which can then use to build proteins, contract muscles, etc. The end product is adenosine diphosphate, ADP, and the phosphate molecule. Additional energy can be extracted by removing a second phosphate group to produce adenosine monophosphate, AMP. When the body is resting and energy is not immediately needed, the reverse reaction takes place and the phosphate group is reattached to the molecule using energy obtained from food. Explain the role of ATP in muscle contraction. Phosphorylation of a protein equals a conformational change in that protein. Skeletal muscle structure, review. Actin, myosin, tropomyosin, troponin, Ca++, ADP, and ATP. http://www.youtube.com/watch V = Kinxine AT, sliding filament theory. Describe the resynthesis of ATP by the ATP-CP system. Creatine phosphate, aka PCR, is a high-energy molecule that is broken down to provide energy for the resynthesis of ATP that is being utilized during the initial stages of exercise. Serves as a rapidly available reserve of high-energy phosphates in skeletal muscle and the brain. PCR and its role during intense exercise. Cannot be used directly to drive muscle contraction. PCR plus ADP plus H-creatine plus ATP. Provides energy quickly but it is short-lived, 20 seconds. When typically used. When performing explosive short activities. Sprinting, explosive weight training lifts, shot putting, high jump, periodic in-game movements, etc. What does your body do once your PCR is used? Lactic acid, lactate, fermentation system. We've seen this before. Glucose pyruvate lactate occurs in the cytoplasm. It's an anaerobic pathway. PCR is 2 equals no O2. Optimal for high energy demands of intense exercise since the energy is produced quickly. However, like PCR, it provides only a short supply of energy, ATP, a couple minutes max. Also lowers muscle pH. Lactic acid is a moderately strong acid, equals reduction in muscle's ability to contract. What energy system kicks in past 2:30 of intense activity? Aerobic cellular respiration of glucose and fat, beta oxidation of fatty acids. We also get ATP production from our fat consumption and fat storage via beta oxidation. Explain the phenomenon of oxygen deficit and oxygen debt. Oxygen deficit, when oxygen need and oxygen supply do not match during the first moments of exercise. Oxygen debt, now better known as excess post-exercise oxygen consumption. EPOC, during recovery from exercise, oxygen consumption continues at a greater rate than needed at rest. Key points of EPOCO2 consumption remains elevated after exercise. 1. To rebuild ATP and PCR stores in the cells. 2. O2 is borrowed from hemoglobin and DSP myoglobin during initial stages of exercise and must be repaid. Oxygen cost to help offset increased body temp following intense exercise. Discuss the characteristics of the three energy systems and their relative contributions during exercise.